Julie Buckler, one of the co-organizers of this conference and part of the Academic Ventures faculty team at Radcliffe. And this is our final panel of the day. We've had a really packed day and some remarkable presentations. But I think this final panel, I'm thinking of it myself as a kind of a scholarly cabinet of curiosities in its dazzling diversity. I think you will be dazzled. One of the questions this panel asks is, what is a collection? A set of objects gathered and subjected to some kind of principles of organization and categorization, representing systems of knowledge and taxonomies, or objects that are grouped because they are somehow the same yet also different, or a kind of an organic whole that has grown in its own way and gone through its own characteristic stages. And one could ask, what does becoming part of a formal assemblage, like a collection, and the etymology of the word collection is assemblage, what does this do to change the status of an object or the context in which we view it? And also, what are the implications of the university as a collector of remarkable single historic objects versus remarkable deep and broad collections? And that's the title of this last panel, Objects and Collections, Remarkable Case Studies. So I'm going to follow the lead of my colleague, Yukio Lippert, and introduce the speakers very briefly in reverse order. And then we'll get to our first speaker, who is a virtual presence. So the final speaker will be James Hankin, who's the Alexander Agassiz Professor of Zoology, Curator in Herpetology, and the Director of the Museum of Comparative Zoology, as well as a Professor of Biology in the Department of Organismic and Evolutionary Biology. Before him, we'll have Professor Jeffrey Schnapp from the Department of Romance, Languages and Literatures and Comparative Literatures. Professor Schnapp also is the faculty director of the metaLAB at Harvard and the co-director of the Berkman Center for Internet and Society. Before that, we'll hear from Francesca Bewer, research curator for conservation and technical studies at the Harvard Art Museums, a conservation specialist. And our first speaker will be Peter Galison, who is the Joseph Pellegrino University professor and the director of the collection of historical scientific instruments at Harvard. Peter Galison was unable to be with us today, but he has provided us with his 10-minute presentation in the form of a film, which we're now going to watch. So think of this as an object in a collection. And you'll see it up on the Radcliffe site along with the other presentations within a couple of weeks. So let's have the film.
Hello, I'm Peter Galison. I'm the faculty director of the collection of historical scientific instruments here at Harvard. This is a collection that began, in one sense, in the 17th century as a Wunderkammer, a chamber of wonders, special, bizarre, strange, illustrative objects collected from around the world. Then, in the 18th century, tragically, it burned down. When the collection was started again as a modern instrument collection, one of the chief initial collectors of these instruments, of electrical devices and optical devices and other things, was Benjamin Franklin, who, when he was in Paris and London, helping to establish the international relations of the United States, was also collecting instruments from the great instrument makers of Europe. And they became the foundational physical cabinet that was used for teaching and research here at Harvard. One of the things that's happened over the years is that the instruments that became part of the research efforts in physics and chemistry and related topics were brought into the collection as a way of storing them for use and eventually for safekeeping. There were also a great variety of instruments that were used for teaching. We have dozens and dozens of binary microscopes, for instance, different kinds of telescopes, all sorts of timekeeping devices from all around the world. And these form a kind of physical residue of the knowledge that was produced and disseminated at the university. What I'd like to do in the next few minutes is to look at three objects that in some ways illustrate the 18th, the 19th, and the 20th century history of this remarkable collection. We have about 20,000 objects in this collection, but without doubt, the most remarkable one of all, the most famous of all, is this 18th century grand orrery. It was finished in the 1770s by the Boston clockmaker Joseph Pope, and in it you can see the planets and moons of the planets as they were known at the time, as they moved around the sun, turned by a set of elaborate gears inside the hole. You see around this lower shape three figures: Sir Isaac Newton, Benjamin Franklin, and Governor Bowdoin, then the governor of Massachusetts. These figurines were cast by a rather well-known figure, Paul Revere. And if you peer through the windows, you can see the mechanism itself. This was not a hidden, obscure, strange kind of mechanism that tried to imitate magic. This was a perfect Enlightenment object, one that would convey the rationality of the universe. Just as God was the clockmaker of the universe, so we could make a model of that clockwork and actually see it turn as it drove the planets and moons along their course. Because so much of what scientists hoped for in the 18th century was embodied in this grand orrery, it's the perfect place to begin our tour through the collection. This is our second stop on this rapid-fire tour of the collection. This clock was up at the observatory about 1.5 miles from here on Garden Street, and it was set to the apparent position of the stars as they moved in the night sky. And from this clock, you can see these two screw mounts on the side to which cables could be attached that would go down the telegraph lines that were owned by Harvard to downtown Boston and then distributed out over all the train tracks that went all over the country and all over New England to set clocks in the stations along those paths. And from the stations then could be used to synchronize clocks in the villages that were there. Now, why was it important to synchronize clocks along the stations of the train tracks? It was because many of these trains only had a single track, and trains would go in opposite directions. At a certain time, one would shunt off to the side so the other could pass. If the conductors had wrong times on their watches, they could actually crash and kill people, which in fact happened in the 19th century. So this was a life-saving device. It was also a matter of convenience. That is to say, if you were in a station, would you use the time of the station, or would you use the time where the train began its trip? So in fact, the first time zones in the United States were set by the train station of origin and would be a time zone 3 feet wide and several thousand miles long. It mattered to coordinate time, and the competition was ferocious to be the university or observatory that would set those times. In fact, this debate over how to coordinate clocks down these train lines became so important that Albert Einstein, when he began his famous paper in 1905 introducing relativity to the world, he began by saying, what do we mean by coordinated clocks? What do we mean when a train arrives in a station in front of me and I say it got here at 7 o'clock? What do I mean if it arrived at a distant station that it arrived at 7 o'clock? How do I coordinate these clocks? And then in introducing the way of synchronizing these clocks, Albert Einstein transformed our modern sense of space, time, and simultaneity. Here we are at the third instrument I wanted to introduce today. This is the control panel of the Harvard cyclotron, almost exactly as it stood the moment it was decommissioned in 2001. We couldn't, of course, save the cyclotron itself. Not only was it huge, filled with radioactive concrete and other toxic substances, but it was simply impossible to even imagine keeping a site that's so important for other purposes now in the biological sciences. But here we took the control panel, and in it you can see a kind of material palimpsest of all the different epochs of physics at Harvard. The first cyclotron was built before World War II and was moved lock, stock, and barrel to Los Alamos to help make the atomic bomb. The government promised Harvard that after the hostilities ended, that they'd rebuild a new and better cyclotron, and they did. This became the control panel for that apparatus. It was at the forefront of physics for a good long time, and it had its ups, extraordinary discoveries across many domains of nuclear and particle physics, and its downs. There was a huge explosion of a bubble chamber in 1965 that caused a fatality and several serious injuries. But here you can see switches and toggles and meters from the 1940s and 50s, oscilloscopes from the 60s and 70s, knickknacks that were used to cheer up the people that were working here. In fact, it's a kind of record of all the different technologies that went into running what was then high-energy physics over a very long time. And when finally the cyclotron could no longer compete with much bigger instruments elsewhere, it became a crucial instrument for medical physics. Its proton beam could be focused down very finely to be able to treat, for instance, tumors of the eye or the brain without damaging healthy tissue nearby. So for a very long time, this was a crucial instrument in the scientific arsenal of Harvard. As we look towards the future of this collection, how we're going to think about these objects in a new context, we, of course, continue to acquire objects, especially instruments that are used in advanced scientific work here at the university. But it's also an opportunity to think about how a university will function with the collection and to put it to new uses. We constantly assemble teams of students, faculty, visitors, to think about each of our major changing exhibits. We did an interesting exhibit, for instance, on the history of the Rorschach test and how it emerged out of other forms of psychological testing and what roles it's played, the debates that have surrounded it. A very interesting show not long ago on the Turing test. This idea that Alan Turing had that if you could make a computer communicate with a human across a teletype in a way that was indistinguishable for a human, that they were talking to a machine, not a person, well, then you would have achieved true artificial intelligence. We set up imitations of that imitation game, simulating the computers and programs of different epochs. And it was a tremendously exciting show. We even had a play written by Stephanie Dick, one of our graduate students here, who assembled the exchanges between people and machines, and between Turing and his parents, as a way of illustrating the complicated dynamics of communication. As we look towards the future, we'll have many other shows. We're planning one now on radio. We're thinking of others about radar and other things. It's very exciting to have this collection, these objects, that can be used not only to recall where Harvard has been in the past, but how to rethink the role of a museum of material objects and knowledge as we look towards the future.
So that video very nicely raises some of the themes that we hope to explore further with this panel, which have to do with reanimating various objects in Harvard's possession, reviewing them, and understanding them anew as they meet a new historical context, which is to say our own. So next, I'd like to introduce Francesca Bewer. The title of her presentation is Treasures from the Long Road in China.
Thank you. I'm honored and happy to be here with you, and also would like to thank all of my colleagues at the Harvard Art Museums for their support in this project and many others. So what I would like to do today is to talk about a small group of objects that were brought back in the mid-1920s by Langdon Warner from the first China expedition. And these are just two representative fragments of wall paintings that he brought back from the very famous Mogao caves along the Silk Route, Buddhist caves from the Tang period. And also this beautiful kneeling bodhisattva figure. Very fragile pieces that are made on unbaked clay or mud painted, and so you can imagine what it must have taken to move those things from the middle of, across the Gobi Desert all the way back to our illustrious institution in 1924. And here you can see a map of where Dunhuan is. And this was a very important sort of intersection of various silk routes and a place of Buddhist pilgrimage. So there were hundreds of beautifully decorated cave temples along this route. The Harvard expedition was initiated under the directorship of Edward Forbes, who was the second director of the Fogg Museum. He came on board in 1909, and he was really the first to start collecting genuine works of art. And as opposed to the collection of plaster casts, this is the museum as it was at the time, and some of the copies in the collection. And fast forward a little bit, but still in the same building before the previous building before this renovation. And by acquiring genuine works of art, he almost immediately started running into conservation issues, especially with the big Italian panel paintings. So he became almost immediately very concerned with questions about the physical nature of the works of art and how to, not only was he curious about how they were made and wanted to learn more about the artists and the production and to share that with his students, but he was also very concerned about preserving these for posterity, and so what could be done about it. And he really was the one who initiated the Fogg Museum in the direction of conservation, and he created the first laboratory, scientific laboratory in an art museum in this country. And the Strauss Center for Conservation is heir to that. So this is just to give you a little bit of context too about the collecting of Chinese artworks at the time. Already beginning in the early 20th century, a lot of other institutions in Europe and also starting in the United States had been traveling to remote areas of China, Asia, and bringing back a lot of material, in the belief also that there was not much interest on the part of the Chinese in their cultural heritage. And because it was such a big country and because it was very difficult also to implement any kind of controls, it was relatively easy to do so, when in fact we know that already beginning in 1909 there was some form of regulation to try to protect cultural heritage. So these are just a couple of images of some of these Buddhist caves, and you can see some of the amazing decorations. This is some of the kind of material that that was found also and brought back by Mark Aurel Stein, who was one of the big explorers of the early part of the century, and he brought a lot of material back to the British Museum. And so it's sort of within that context that Edward Forbes and Paul Sachs and people at Harvard felt like they really wanted to contribute to our collection here and enhance it and really enter the world stage as well. And Langdon Warner, who was a Harvard graduate and was at the time a curator, actually the director of the Philadelphia Museum, had also been a curator at the Museum of Fine Arts, was a friend of Forbes. Anyway, he was brought on board to direct not only this expedition, but also the second expedition. So he set off with a very small group, and in fact his main partner fell ill and was sent back, so he ended up on this solo trip. He originally was planning to go to Karakoto and to scout out areas for possible archaeological explorations, and decided, since most of that had been cleaned out by previous explorers, to continue to Dunhuan, and ended up in these amazing cave temples. And this is a photograph that he took of the cave, and you can see our kneeling attendant bodhisattva here. This is a picture that he took before, and this is taken shortly after its removal from the cave. And this is a modern day picture of it. And these objects are the source of a bit of, I think, anxiety at the university, because the Chinese have been very, what's the word? felt very strongly that Harvard pillaged their cultural heritage. And so, anyway, something we might talk about later. So in preparation for the expedition, Langdon Warner turned to Edward Forbes, who he knew was very interested in conservation issues, and asked him for advice on how he might remove wall paintings if he ever got a chance to. And Edward Forbes consulted with his assistant, Daniel V. Thompson, who's better known for his translation of Cennino Cennini's treatise, 15th century treatise of the Handbook of the Artist. And he had spent a lot of time in Italy and had talked with restorers and knew a little bit about the transfer of wall paintings. And there are two different ways of transferring wall paintings. One of them is to cut a chunk of wall with the paint layer out. So you have the substrate with it. And the other, but that sort of limits the size of the chunks that you can cut out. Or you can use a technique that was called strappo, which was developed in Italy, which is illustrated here, which is basically very similar to depilatory waxing, where you put a layer of facing, glue a facing on the wall painting and pull off just the pigment layer. And this is what this famous restorer Stefanoni is doing here. They sort of consulted his work. And there's one whole chapel that Stefanoni removed in the Museum of Fine Arts today, and some of you may recognize it. It's a very interesting story as well. So with some information in hand, Langdon Warner set about removing a selection, actually 12 fragments from the walls in some of the most important caves on the site. And you can see here, this little darkened area is where he had started applying some of the fixative before he applied the facing. And this is this fragment here. This fragment here. This is the only picture that we have from him of a sort of treatment and this is a more contemporary picture of another site where he removed some of the fragments, and you can see where it really just leaves a bald spot. And here are the two fragments in the collection. Now, the problem is that the strappo technique was developed for fresco paintings, and so when you stick glue on a fresco painting where the binder is really it's a chemical binding with the wall itself, with the lime plaster, it's not that difficult to remove the glue afterwards. But in the case of these mud wall paintings that weren't fresco, where the binder was glue, and with the inexperienced hand of Langdon Warner, and he describes his difficulties in this. I mean, he basically caked the surface with glue. It was incredibly difficult to remove the facing afterwards. So we have on display, I think, 4 or 5 of the ones in better condition, but there are a number of fragments that are behind the scenes, which are more useful for study purpose at this point. The whole idea was really, I mean, he was convinced that these sites had been abandoned, that they had been damaged, and that he was really doing something to preserve at least a remnant of them for, the Western world. And he actually describes in a letter back home, he sort of compares the condition of these sites to Louvain, which was one of the famous sites of hecatomb during World War I. There's a very good article about this whole history by one of our former interns, which I recommend if you're interested in the history. Fast forward a little bit, Dan Thompson, when these fragments were brought back, had a very hard time doing much with them, but he did what he could. But shortly thereafter, Edward Forbes created the first conservation department in the museum, and this is George Stout, who became the first conservator, And Rutherford John Gettins, the first scientist, really one of the first tasks that were given was to tackle this problem of what to do with the existing fragments, but also how to develop a better way of removing and transferring frescoes, and especially mud wall paintings like these. And this is just some of the material that we have. They really introduced a more scientific approach. We have no records really of what Langdon Warner did. They also developed a kit for Aurel Stein, who they hoped would go on another expedition for Harvard and bring some back. I'm skipping a whole episode of a second thwarted expedition. They were thwarted in their attempts to bring back entire chapels, much like Stefanoni had done. But as a result of having done this scientific work and presenting it at several very important European meetings in 1929, 1930, one was the Congrès International d'Histoire de l'Art, and another one was the first international meeting on scientific examination and restoration of works of art organized by the International Museums Office. It really propelled the FOG into the center stage of the sort of budding scientific conservation field. And really that is what I wanted to emphasize, that even though these objects might not give us the full glory of the art that they represent in these caves, that it did sort of push us in this into this amazing role in the history of conservation. And they developed techniques, and the use of materials like polyvinyl acetate was used for the first time in conservation by them in application to these murals. This is an example of some of the wall paintings that were sent by other museums later on to the FOG to have them conserved. And you can see an example here of what those mud blocks looked like when they cut out the chunks rather than ripping off just the fragments. We ended up, Gettins and Stout and other people at the museum ended up creating one of the most important early arenas for exchanges about conservation issues, technical studies in the field of fine arts, and also ended up being among the founders of the International Institute for Conservation and the AIC, which is the American version. And I just wanted to end by saying that we have had, with the renovation, the opportunity, the FOG renovation, the opportunity to really revisit some of these objects more closely, and especially the kneeling bodhisattva, and to be able to do the kind of technical analysis that even the Getty Conservation Institute and the Dun Huang Institute, who have been doing a lot of conservation research on site, can't do. For instance, I'll come back to this in a moment, X-radiography, because that's something that's more difficult to take into the field, which has allowed us to really see what the internal structure of one of these unbaked clay pieces would look like, or how it was made. And we're still in the process of doing scientific investigations on the pieces. It's very exciting to be able to finally get our hands on him. And we're hoping also to collaborate with the Dun Huang Institute. Thank you.
Thank you very much. Thank you, Francesca.
Good afternoon. I'm going to touch upon a number of themes that have come up in some of the earlier sessions and presentations. And one of the sort of overriding questions I want to ask is this question with which I lead, which is, the world is filled with collectors and collecting institutions. Collecting is an infinite task. And so I want to ask some questions about to what degree we can think about university-based collecting, universities as collectors and as stewards as a distinctive function vis-a-vis all of those other agents of collecting that take place. And I'm particularly interested in this notion, which I think is a deep part of the history of many of the areas that characterize certainly Harvard's collections, which is the fact that they were born as working collections. They were not born as efforts to collect per se. Many of them were born as direct outcomes of efforts to support and serve the production of knowledge as part of research and teaching, whether that's understood in a kind of immediate way or it's understood in a long-term way. So what is a working collection, I guess, is the question that interests me here. And how are working collections different from ordinary collections? What kind of artifacts are the distinctive, defining kinds of artifacts that make up such working collections? And the example that I've brought for purposes of today's conversation, I hope falls under this broader category that somehow it seems to me that one of the things that could be an appropriate answer to the question of what's distinctive about university-based collecting, or what should be distinctive perhaps, is its special attention for unlikely candidates for conventional collection and conservation. Objects that have either some degree of ephemerality to them or endangerment, or that are entangled in ways that create artifacts that tell stories, sometimes extremely densely woven together stories, but they're stories that are not necessarily defined as possessing value by capital or by the kinds of markets that define many practices of collecting. And instead, they have the possibility of embodying forms of value associated with process. Process can mean a lot of different things here, but I want to at least explore the idea, an idea that would be a capacious concept of conservation, of collecting, that would include, for example, the plaster casts that were central to artistic training, as well as archaeological work in the 19th century. So my example of such an ephemeral, endangered, or entangled object, or one that crosses my path regularly, is going to be a model, an architectural model. As many of you may know, the practice of architectural training, like most design fields, is very much a kind of translational practice that moves between theory, history, and all kinds of forms of making, including model making. And models have always been a real problem in architectural history. I teach across this FAS and GSD divide, and I'm always struck by all of these models being dismantled as every academic semester ends. Some of them are very beautiful. Others probably are not worthy of preservation. But the question of what is a model as a kind of object? What does it mean to collect models? What kinds of things do models do? Can they do? How can they animate a process rather than just be treated as products, is the question I'm interested in here. And I want to sort of, as a framing question, think about this degree to which we have remained true in our own collecting practices to a vision that thinks of collections largely as having value as a function of their ability to sustain and support forms of knowledge making. So, as anticipated, the object that I have chosen for today's conversation is a model. It's a model that you will encounter as you walk down the stairs into a Loeb Design Library. And as it's encased in a nice plexiglass case on which you will find the following label. The Total Theater, designed by Bauhaus director Walter Gropius for the director Erwin Piscator. If Piscator isn't familiar name to you, it's worth knowing that he is one of the truly revolutionary figures in the history of the experimental theater. He's a socialist, and he imagined new forms of spectacle that would integrate cinematic projections with live embodied forms of performance on a very large scale. So within this framework, we're told by the label that accompanies this model, the 1927 Total Theater was envisaged by its designer as a variable theater machine. And it goes on to emphasize some of the features that I'm going to come back to in a minute. It was never built, and then it concludes by noting that this model was built as a component of the 1992 exhibition, Walter Gropius's Total Theater design of 1927 for the Busch Reisinger. Now, despite the lovely Herb Lubalin-inspired typography here, the Total Theater was designed by Gropius, but in fact, he had a collaborator. This label has at least 4 imprecisions, perhaps even distortions to it. that collaborator was a specialized theater designer named also a Bauhäusler, Stefan Sebuk, a Hungarian architect, and it was inspired by the work of many others. This project comes late in the history of theater experimentation at the Bauhaus. The second is that this model is a replica of a lost original. And indeed, it was made for the Busch-Reisinger, but it essentially takes that original, that lost original, and adds a series of mechanical enhancements and interpretive changes in the model itself that I want to talk about. Third, the model was not built for the exhibition at the Busch-Reisinger, as I'm going to show you. It was built for the US Institute for Theater Technology in 1973, with financing from the National Endowment for the Arts, along with a personal investment by Isa Gropius, Gropius's wife, as part of an exhibition effort to bring together the two portions of Gropius's career, the Bauhaus, the German portion that goes up through 1937, and the American portion, to bring them together and to show the continuity between the great projects, whether they were built or unbuilt, of the Bauhaus era and the work that he was doing in the United States after his immigration. And it's worth mentioning in that same regard that this model, the Total Theater Project, was literally the passport that got Gropius out of Germany as the Nazis rose to power. As I'm going to document, he actually the last presentation of this project took place in 1934 in Rome in the context of a major international congress on the future of mass spectacle forms connected to, that would somehow bring the theater and the cinema together to compete with rival forms of spectacle like stadium-based mass athletics and the cinema in particular. In short, this is an object that has a really interesting layered kind of history that includes peregrinations through many different entities at Harvard at the time that it finally makes it to Harvard from these various tours. And last but not least, there's a piece of direct family romance in here. This model was built by Gropius's son-in-law. It, in a sense, continues Gropius's legacy literally as a kind of family endeavor. It isn't just Isa Gropius who's engaged in promoting this kind of unified vision of Gropius's career through the construction of these kinds of artifacts, but it is also his son-in-law. So Gropius, of course, had a relationship to Harvard that was an intense and deep relationship. He was chair of the Department of Architecture from 1937 when he arrived in the United States to 1951. Most of his archives, at least his American archives, are held at the Houghton Library. He designed several campus buildings. The Architects Collaborative was a key player in the Boston area in the dissemination of certain ideas about modern architecture. We have a large collection of drawings, including the drawings that document the Total Theater Project at the Harvard Art Museums. And last but not least, in 1992, the moment that this model comes to Harvard in a sense, a permanent sense, was an exhibition that was held at the Busch-Reisinger, devoted to Walter Gropius's Total Theater design of 1927, a variable theater instrument. So we have the materials in the archives, in the Harvard Art Museums, including all the drawings of the project. And we have this model, which currently is located, as I said, at the Loeb Design Library, but that goes through a very complex chain of command from between 1992 and the time and the present time. So here we have an image of the original model, and you'll notice that that original model has a couple of major differences. It was a wood and plaster model. It was a prototype that was intended to elicit the financial support that would have been required to build Piscator's theater, a kind of theater machine with all of the trappings of what multimediality was conceived to be circa 1925. It had projection systems from all sides, 3 rotating stages, inner and outer circles that placed spectators in a constantly fluid relationship to the actors. And moreover, it was itself conceived of as a place where spectacle forms that would cross the boundary line between the cinematic, the athletic, and certainly the political would all intersect one another in a new kind of spectacle factory. It was a project that was building on the work of many, many people, But you'll notice in the model there's one difference that you see right away, which is there are actually dancers on one of the stages. And not just any old dancers, but they're dancers who come out of the fourth Bauhaus book of the work not of Walter Gropius, but of Oskar Schlemmer, a fellow member of the Bauhaus who was very actively engaged right from the early 20s, long before Gropius became involved in stage design, in imagining new forms of theater and spectacle. And alongside Schlemmer, we find figures like Moholy-Nagy, Marcel Breuer, Zanti Shavinsky, Farkas Molnar. They were the true protagonists of the development of ideas around the theater. Gropius's work comes along and, in a sense, consolidates them. What the model does is to weave together this story that was, in fact, separate threads that really didn't come together in the Total Theater project, but that, in a sense, kind of seal historically the connection between Gropius's work and the work of a whole community of people. Here we have a slide of one of the most famous projects of Schlemmer's from the period, the Triadic Ballet, which was performed variously between 1922 and the time of, through the early 30s. So I mentioned that the Total Theater project was literally Gropius's passport out of Germany back first to the UK and then to the United States. And in 1934, at the Volta Congress, he presented this project for the last time until the exhibition, the touring exhibitions of the 1970s with the model that whose life we've been tracking, as this sort of total theater machine. And it's important to remember that this is in the context of a theater congress where we have Meyerhold, the great Russian experimental director, in place, and we also have the protagonists of fascist Italian experiments with mass total theater machines with similar kinds of inspirations involved. This was a conversation that was felt really to be trying to ensure the survival of the theater, competing with these other kinds of cultural forms and forms of spectacle. So to go back to the genesis of the model, the US Institute for Theater Technology commissions in the year 1972 Charles Forberg, Gropius's son-in-law, to build this model for the purpose of a series of touring exhibitions. And indeed, those exhibitions take place, and we have subsequent correspondence in the archives about this device, the maintenance. These are the instructions from Charles Forberg from the Harvard archives. And you can see that it not only adds an interesting interpretive twist, which is the presence of these dancers that are borrowed from Oskar Schlemmer's work, but it's also a mechanical model, unlike the original model. And the choice of a mechanical model, a model that has a whole series of spectacular mechanical capabilities, including a lighting system, as well as these dancing figures, I think is really powerfully inspired by the desire to place the Bauhaus's experimental work on the theater at the root of the tree of emerging forms of immersive spectacle in the 1970s. This is just a slide from Stan Vanderbeek's Movie Drome, which is just one of the many, many projects in this very period that are experimenting with immersive models of spectacle. So to fast forward here to the end, we reach in, after this object has existed, circulated in exhibitions, it ends up in the warehouse of the Architects Collective for a number of years. It gets transferred to the Busch-Reisinger in 1992 for this exhibition, which puts all of the pieces together that are now at Harvard. That exhibition takes place, and then the question becomes, what's going to happen? This is a big, messy model. It requires a lot of maintenance. It breaks all the time. It's got too many bells and whistles. And so the USITT persuades the Busch-Reisinger to acquire it, and a series of migrations take place, first into the theater archive, then eventually to the GSD, and maintenance takes place on various occasions. The last occasion of maintenance was when I taught a course about 3.5 years ago. One of my students became interested in this model, and by the way, it's only one of dozens of visionary theater projects that are enshrined in the theater collection. And in the context of that course, the student's project became a restoration of the core functionality of the total theater as part of a kind of prehistory of ideas about immersive spectacle. But we have many other such models. This is the Universal Theater of Kiesler, which is a project that is born in the 1920s and then is resuscitated in the 1970s and so forth. So here's our icon of the kinds of interventions that such structures require. These are messy, complicated objects, but they're objects that are very rich in stories. And I want to close here by poking a little bit of fun at the Gazette. So they covered this event as the things Harvard has. And I think that gets it exactly wrong. That I think what we ought to be thinking about is the things Harvard does with the things that it has. And that means that we need to reflect upon the kind of horizons of the sort of collecting practices that we engage in. I think in a way that goes beyond the sort of typology or the concept of just sheer accumulation as a kind of social good. Thank you.
Thank you.
Thank you very much. Let me begin my presentation by asking a question. Don't you have anything better to do on a Friday afternoon? Thank you for sticking around. This subtitle for my talk could be, and now for something completely different. Because I come from a different world, as you can imagine, than most of the presentations we've heard today. And that is the world of natural history collections. Harvard has the largest natural history, university-based natural history collections, university-based natural history collections in the world, of any university in the world. They're truly enormous. They rival in size those of the major freestanding museums around the world. And there are very different imperatives that we face and subscribe to when we develop and utilize our collections, and that's what I'm going to try to convince you of today. And another way I took a different tack is we have, believe me, just in the Museum of Comparative Zoology, which I'm the director, we have more than 20 million specimens. We don't actually know how many, but it's a lot more than 20 million. And some of them are quite spectacular and beautiful and so forth, but I took the different approach, and I said to myself, I'm going to try to choose among the most boring, mundane, matter-of-fact specimens that we have and base my talk around that and show you how, if you treat a specimen like that in a particular way, you can really make it spectacular. To give you an idea of the kinds of things, if we can do this to a really mundane, boring specimen, think of what we can do with an inherently exciting one. So here's the specimen. It's actually sitting in its jar. It's an earthworm. It's an earthworm that was collected in the very exotic locality of 26 Oxford Street, Cambridge, Massachusetts. And in fact, there it is. You can see it in all its glory. We've turned the jar around. There's the earthworm. It's a common earthworm, nothing special about the species. It was collected a couple of years ago. And the locus for all of the information I'm going to talk to you about is shown here. This is a page from our database, museum database called MCZBase. I should also, just before I go on, I should mention, come back to this question of imperatives or topic of imperatives. For natural history collections, there's a tremendous premium on extracting as much information as you can possibly generate from specimens. Information that can be used in scientific research, in teaching, as well, which are the two principal uses, I guess you'd say, of our natural history collections. But I think, as we all know, there's a tremendous interest in natural history collections by people from a variety of other disciplines. We get a lot of use of our collections by illustrators, by artists, by conservationists, by grade school teachers, and so forth. And we want to extract as much information as we can, and we also want to distribute and make the information accessible to the maximal ability by as many people as possible. Anyway, back to MCZBase. So here's our specimen page for that earthworm. There's the scientific name for it up there in the upper left. There is the locality, 26 Oxford Street. That's where it is. Here it was collecting source, wild caught. So you can just imagine the drama that unfolded there on Oxford Street. And you'll notice, in addition to the locality, it has these coordinates. These are georeference coordinates, latitude and longitude, which is more than a desiderata, it's absolutely mandatory these days when people, for reasons I'll show you in a moment, when people collect natural history specimens, we don't just want to know where it's from in terms of the address, even if it's a very specific address, but we want to have latitude and longitude. And that's for a very simple reason. If you have latitude and longitude, you can generate instantly, in real time, maps such as this one up there. You can pinpoint anywhere on the globe where your specimen comes from. So if you were to click on that little map in the upper left and enlarge it, is it possible to lower the lights a bit, the spotlights? This is coming out very light. That's a map from Google Maps. It plots the locality of the specimen on Google Maps. So there's our museum on Oxford Street. By the way, we're over here. There we are, up here. And this is a very mundane example of this, showing for a specimen that's collected here in Cambridge. But we can use the same principle to generate in real time the location of any specimen collected anywhere in the world, no matter how remote. And you can do this for 1 specimen, 10 specimens, 1,000 specimens, as many as you want. It takes a microsecond to generate and get immediate range maps and so forth. Extremely important. You can then do overlays of satellite depictions of the terrain and so forth. Just that in of itself is a tremendous asset. Now let's go back to our MCZ-based entry for this specimen. Over here in this section of the page says media. And another thing, obviously, for a number of us who are doing it, we want to put digital photos of our specimen online so people can see them. And I'll just show you, here are 3 static images. I'll show you one of them here. For those who've really been curious about what the guts of an earthworm, there you are, and they're already labeled. Now that satisfies one imperative. It provides a lot of information to people who are interested in this specimen and what it looks like, but it also contradicts and defeats another very important imperative of museums in general, which is conservation. And preserving, one of our mandates is to preserve specimens intact indefinitely. We're not supposed to do any harm to them except in particular circumstances, particularly situations where the circumstances dictate otherwise. And so that's why another kind of imaging is being embraced by natural history museums. And those are the images that are represented here. I'll show you enlargements of these in a moment. And these are images produced by a very sophisticated, elaborate X-ray technology, which is now increasingly widely used, called microcomputed tomography, and this is called micro-CT. And micro-CT, as you can imagine, is a non-invasive, non-intrusive form of imaging, not only the outside of materials, but the insides with tremendously high resolution. Now, I'm going to back out of PowerPoint for a moment just to show you a couple of our images. So here, for example, click to activate. There's our earthworm that we put into the micro-CT scanner. And this, I should say, is a 3-dimensional PDF, which you can download from that page, that MCZ-based media page, onto your desktop. And you can do all of these manipulations at your leisure. We were just looking at the standard view. There's the mouth. Here's the standard view transparent. Now that was produced by the micro-CT without damaging the specimen in any way. It takes advantage of the fact that all the guts, all the things inside the specimen, react differently to the X-ray waves, and they're interpreted by rendering their shapes exactly as they are, and their relationships of all the different organs in the body to them exactly. And you can even take this and do some more complex, let's go up to the digestive tract. You can pull out the guts, for instance, and we've labeled them here. You can actually, here's the 3-dimensional part, you can manipulate it yourself, you can turn it around, you can look at it from the front, you can look at it from down, you can turn it over if you want, and so forth. You can also dig in on other organ systems in increasing detail. For instance, we're looking for the nervous system here. Now, those of you who can look or have good eyes can note there's a yellow tube on the underside of that animal. That's actually the nervous system, that's the spinal cord. And we can then gradually remove other parts of the body so you can see it increasingly effectively. And there's the nervous system. We've literally virtually pulled it out of the animal fully intact. And then you can actually look at the brain and label it like this. Now, just if you're curious, there actually are 2 micro CT scanners on campus. We have one of them in the museum. There's another one across Oxford Street at the laboratory, at the Center for Nanoscale Systems. If those of you who are not familiar with it are curious if it works for other specimens of inanimate objects and so forth, the answer is absolutely so. I can just tell you as an anecdote, one of our postdocs a few years ago helped the Harvard Semitic Museum do a CT scan on one of their Egyptian mummies. Not a human mummy, but Egyptians, as you may know, mummified a lot of animals. And they have a bird mummy in the Harvard Semitic Museum, and they were curious what kind of bird was it, what did it look like, and so forth. And one of our postdocs prepared a beautiful CT scan of from their mummy, which I think may be available on their website. Now, one can take this even a little further and take that CT scan and start moving down from one side, in slices, from one side of the animal through to the other, as we're doing here. We're basically moving, we're looking at a 2-dimensional plane, but we're moving that 2-dimensional plane down through the animal. Now let me see if I can turn this a little bit here. Can't get to my, well, okay, we're coming back through I'll just let it run a little longer so you can see, because it's going to rotate in a moment. We're coming up through the animal again. I can explain to you how CTs work. CT produces a series of 2-dimensional images, but it does almost an infinite number of them, and produce the 3-dimensional rendering. It stitches together all of the sequential successive 2-dimensional shapes and gives you the 3D animation. So now here we're rotating the specimen. We're turning it on its side. And now we're going to start going through the specimen again from outside in. So again, these are tremendously informative materials both for research, but also for teaching. And frankly, they're kind of cool to look at, especially from some other animals that you might be a little more familiar with. Okay, let me stop that and go back to our PowerPoint. You might ask, well, why did we do an earthworm? It turns out this is technology that is still being developed, and we collaborated with a couple of scientific journals who are interested in seeing if they could do this and deliver it as parts of publications, high resolution renderings that could be downloaded from electronic journals. And the earthworm was chosen just as a model. Truth be told, there's not all that many people who are really curious in the internal anatomy of earthworms. But if we could get this working properly, we can do this on just about any animal. And that is why it was used, and it's been quite successful. Now let me, just in another couple of minutes, let me mention another thing that is very characteristic of collections for natural history these days is not just the static specimens in jars or dried bird specimens and so forth. For virtually every specimen that comes into the museum these days, and we still get a lot of specimens coming in from fieldwork, we take a little piece of it and freeze it. We really freeze it. We put it in liquid nitrogen at about minus 280 degrees. And we now have a cryogenic collection. Here's a photograph of our cryogenic collection in the ground floor of MCZ Laboratories. And the cryogenic collection, these frozen tissue samples, are then available for researchers who want to do studies of molecular biology and do DNA sequencing. And so DNA sequencing and other molecular approaches are extremely widely used now in natural history collections in the study of evolutionary biology, phylogenetic relationships, biodiversity inventory, conservation, and so forth, a whole range of approaches. Now, here's another species page. We don't have actually a molecular sample from that worm I showed you, so I thought of, well, what's another animal that's probably even more distasteful and disgusting than an earthworm? How about a little spider that looks like a mite, or looks like a tick? And that's what we have here. But the reason and you can see a picture of it there on the lower right. The reason I'm showing this one to you is for a couple of reasons. Here's the name there. And this is a particularly nice illustration because this is a new species of spider that was just described this year in 2015. It was work that was done by a Harvard undergraduate, Taras Dresser. And if you're curious, you can actually go to our site and download, go over here, and you can download the publication that Taras Dresser published naming this new species of spider. But it's up here that I want to show you. See these references here? These are all links. And if you were to press on one of these links, your computer would take you to this site, which is the National Center for Biotechnology Information, where you can access the DNA sequence of this particular gene of this particular animal. There's the species Cyphophthalmus oleitensis, the voucher specimen, that's MCZIZ135079, and you can access the DNA sequence for the cytochrome C oxidase subunit 1 gene, or any other gene that has been sequenced for that particular species. So we're linking out our data to all other sources, knitting together a tremendous information resource that is literally at your fingertips. We're also sensitive to the fact that our information is used a lot in teaching and for biodiversity education in general. There's an enterprise called the Encyclopedia of Life that the MCZ has been very active in for several years. It's an activity that was created a few years ago. We received large grants from the Sloan Foundation, Alfred P. Sloan Foundation, and the MacArthur Foundation to create the Encyclopedia of Life, which, when it's fully developed, will have a web page for every species of living organism, all 2 million species, with as much information about those species as we can assemble together. And here is the web page on the EOL, and this is I should say this is a resource that's used tremendously by grade school teachers. And from the Sloan Foundation and the MacArthur Foundation, as much as they like scientists, it wasn't scientists that they were looking to support. They were looking to increase the biodiversity knowledge in lay people, the general public around the world. And you can maybe recognize or see those images. They may look somewhat familiar. Those are images that are taken from MCZ's website, from our database, and we've now configured our database with EOL. So every time we upload an image to MCZ base, it's automatically harvested and put in the corresponding species page for the Encyclopedia of Life. It's a tremendously versatile system. Again, sharing our information more widely. Okay, my final slide just depicts, wouldn't it be nice, I've been telling you so far, things about MCZ specimens principally, and MCZ uses. Wouldn't it be nice if we could take our information and combine it with like information that is being generated by all of the other thousands of natural history collections around the world? Well, in fact, that means exists, and it's done through a mechanism called the Global Biodiversity Information Facility, GBIF, which is based in Copenhagen, and virtually links together collections of natural history collections around the world. As of this month, there are 500 million records contained in GBIF, which is, on the one hand, a lot, but we estimate there are between 2 and 4 billion records in natural history museums around the world, so we still have a lot to go. And on this map, GBIF has produced, they've taken a tiny red dot for every one of those 500 million records. And you can see where those records that are georeferenced, you can see where they are found and where the gaps are and so forth. So this represents a tremendously exciting activity, but it also changes our view of what a collection is and what a university collection is. We are not just serving, we the MCZ are not just serving an audience here at Harvard. We are serving a global audience. Likewise, investigators here at Harvard are utilizing not just information based here, but we're utilizing information found elsewhere around the world. So instead, what we're gradually moving towards is not a series of individual isolated collections, but a globally linked uber collection of natural history specimens from around the world, all of whom have agreed to embrace the common standards for interoperability, data management, and so forth. So it really is a very different model from what has prevailed up until recently. So I'll stop there. Thank you.
Thank you very much. I would like to just re-emphasize Jeffrey's very important point that what is distinctive about the university as collector is that the university's collections are working collections dedicated to the production of knowledge for research and teaching, and also repeat Jeffrey's excellent formulation that this is about what Harvard does with what Harvard has, not just about what Harvard has. And I think this final example with the earthworm shows the mind-bending implications of where we're headed. So we're officially out of time for this panel, but I don't want to prevent anybody who's here who would like to ask a question of the presenters from doing so. So I wonder if there is someone who would like to ask a question. Yes, I can see that Professor Ehrlich would like to ask a question.
I would love to. They're all very, very interesting, but I can't help but ask Jim, because this was fabulous and accessible and exciting and wonderful. But I'm a historian, and I'm very interested in MCZ is not only one of the biggest, it's one of the oldest. And I'm quite sure your early artifacts don't have the kind of detail that the things you're collecting today do. And yet, from a historian's point of view, they're very important. Environmental history, climate change, biodiversity, all of these things are historical as well as contemporary questions. So I'm wondering how you're thinking within the museum to solve that. Can you make comparisons over time? How can you access 19th and maybe even, I'm not sure, the early, do you have some 18th century things as well?
The great question. Actually, fret not. We tremendously value our older specimens, and in fact, they are part of what makes the MCC so valuable. You'd be surprised, though, how much information they do provide. Now, one of the reasons for animals, they provide extremely important information. For no other reason, many of those species are now extinct. And they represent irreplaceable records, or at least even if the species are still exist, they may be no longer occur in regions where they once did, and they provide exactly that information about historic ranges. You know, you can get DNA out of dried museum specimens even hundreds of years old. It may be a bit degraded. Maybe the sequences are not as good as you like them, but you'd be amazed what you can get. We can generate, maybe I did mention this, even though people when they were taking their localities, they didn't take their latitude and longitude, but we can do that now. There are gazetteers, we can, for most specimens, figure out pretty well where they were found, where the localities. If we can read the handwriting on the tags, we can pretty well figure out where they were, and we can give it a latitude and longitude. So actually, the historic specimens are among the most valuable, and whenever possible, and to the full extent possible, we include them in these analyses for exactly the reasons you talk about.
It does seem that these collections have to be reactivated all the time in order for us to continue to reanimate them through rediscovery and reexamination. And the panel showed us Francesca's talk, for example, and also Jeffrey's, about the need to maintain, preserve, and conserve these items. I see you have a question as well.
Hi, I'm Katie. I'm in the music department, and this is a really big topic, but I'm wondering kind of politically about maybe problematizing what the university is in this title, and problems with space and funding and that sort of thing. We have a collection of musical instruments, actually, that has no curator. It's in this tiny little room. Things are falling apart. So I'm just wondering if maybe someone would speak to what can I, as a graduate student, do. And if anyone here, has an extra 10 million and would like to talk to me after, that'd be super. So thank you.
I mean, I would love to jump in on that one. I think, and this is, by the way, not unique to universities as a problematic, but I think our institutions are very good at accumulating stuff and very bad at knowing how to do anything with all that stuff. even universities. And it isn't by accident that, depends who you ask, but 90% of the cultural heritage of humankind is in deep storage somewhere, and unlikely ever to be exhibited in any form. And so in my own work as working at the frontiers of sort of what I like to call knowledge design, but digital arts and humanities, I mean, I think one of the great areas of opportunity is precisely to use a kind of digital wedge to unlock the power of a lot of those kinds of resources that are not being curated as physical artifacts for a whole legion of reasons. But of course, no matter where you look, there are costs and there are political choices, there are ethical considerations, there's a legion of obstacles to that process. But I think you asked a very also pragmatic question of, what can a graduate student do? I think that understanding scholarship as encompassing curatorial responsibilities and activity is one emerging attribute of it's in a sense a 19th century model that's resurgent under new conditions today, and I think there's an opportunity to work locally with collections that are in front of you and otherwise locked in these kinds of paralytic sort of or logjam-like situations where they have important stories to tell, but they cannot be told because they're not accessible. Or so anyhow, I mean, I think there's a multiplicity of ways to attack that problem, but it's a very important problem that you touched upon.
May I just jump in? I'll answer, try to be brief. I'll answer it in two ways. One, just a general observation I've made. I've been at Harvard since 1999, and I must confess I've been shocked at how badly the collections, museum collections, institutional collections here have been maintained by the university. I think universities should be colossally embarrassed for how little they have, until recently, how little they have done to sustain the collections in the proper manner. I mean, it's the wealthiest university in the world. There's really no excuse. Now, obviously, there are very encouraging signs. Art Museum is probably the paradigmatic example. We in the MCZ, we now have, we're given 50,000 square feet of new state-of-the-art collections space in the Northwest Science Building, so we've been treated well. So there are signs, but there are still many other collections which deserve much better, and it's a problem. The university should be embarrassed. I mean, that's just my basic observation. Specifically, though, about your point, I think you're within faculty, music school, or music is within FAS. FAS, one of my mantras has been since I've been director, FAS has two priorities. One is faculty research and one is teaching, undergraduate and graduate teaching. If you can align your activities with one or both of those priorities, you stand a better chance of succeeding than if you don't. It's not rocket science, but not all units do this. And I think for your collections, you need to demonstrate that the collections are not only used, but they're indispensable. They're critical to achieve the teaching and research mission of the unit. And in my experience, deans are more receptive to that kind of an argument. They are not interested in sustaining collections just for the sake of sustaining collections if they're not being used. I've heard deans, administrators say, well, gee, if we're not using this stuff, why do we keep them? We should give them away. So, and I understand that logic. So we're not a freestanding museum that's just to collect objects. We are a scholarly institution, and the collections are part of the capital of the university that need to be integrated into our academic programs. So,
Francesca?
I was going to say something a bit along the same lines, but I think that, with the digital technologies and ways of making people more aware of what is in the collection, I mean, you could start working on that, with other graduate students or people interested, but also that then makes it accessible to people out there who are interested in that kind of material, who would want to collaborate with you and use them as working collections and lend them more value. So I think we're in a time when that is all very possible, as we have seen from the examples here.
Yeah, if I could just piggyback on that, I think the real horizon that we should be working towards is one where we, first of all, understand that objects are themselves collections of attributes. I think James's examples were wonderful in that regard. In other words, that every object is already saturated with sets of networks of relations and so forth, and that collections need to talk to other collections outside of the orbit of a given collecting agency, because it's when you start to build those collections of collections of collections that you get into this vertiginous territory where all kinds of possibilities open up that go far beyond the kind of limited horizons of a given collecting institution. And I think for an institution with Harvard's traditions, that's a difficult leap to make, to start to think of yourself as just one pole within a kind of network of collecting entities that are all operating in this sort of open, interoperable, open, resource environment. But there's a tremendous amount to be gained for scholarship, for the kind of rigor of the work we do, the kinds of stories we can tell. And I really think that, particularly for the historical fields, this is a really important horizon.
And also thinking of the materials as being really interdisciplinary, objects of interdisciplinary interest. So not just for, one particular field.
I was at the Schlesinger Library. I retired when you arrived. You mentioned...
hopefully not because I...
I think there's no causal connection either way. But you mentioned, in response to Laurel's question about being able to read the tags, we have students now, I understand, who cannot read handwriting at all. And so I think that has implications across the university and across all these collections, not just at the Schlesinger Library, where, the bulk of this stuff is in 19th century handwriting. So I just wanted to mention this as a sign of the times.
It's a big problem for it. We're actually working with computer scientists who are developing smart systems to translate handwriting on tags, just like banks do it on checks and the post office does it on addresses on envelopes. So ultimately, somebody has to be able to read it to correct the machine, but your point is well taken.
So let me just conclude very, very briefly. Certainly there are many challenges that we face, but the increasingly object-centered teaching that's being embraced across the disciplines is very promising. And with the materialist turn in the humanities in fields like mine, literary studies, scholars are increasingly using objects as narrative devices for telling diverse histories, making the objects speak. So we hope this symposium has reflected a sense of renewed mission that's in the air, a coming together of specialists based in collections with scholars based in schools, departments, and programs. And with all of the resources and expertise and the new tools and technologies at hand, there really is the potential for a profound intertwining of research and teaching that can be transformative for us and for our students. And that will also allow us to reach out well beyond Harvard's walls. We're very appreciative that you joined us today, that we were able together to explore some of these possibilities. We hope this symposium is just one of the events going on around Harvard these days in the air that will help jumpstart some new productive collaborations to add to those that are already springing up around Harvard. I would like to conclude by giving my sincere thanks to the Radcliffe Institute for sponsoring this symposium, to the librarians at the Schlesinger, Sarah Hutchin and Amanda Strauss, who created the pop-up exhibit, to Becky Witham and all of the staff who made this event run smoothly, and particularly to Rebecca Wasserman for her guidance, her advisory role, her enthusiasm, and her investment in the intellectual goals of this conference. If you've enjoyed this event, you might also enjoy the Rosetta Elkin exhibit, Live Matter, that's opening here on May 5th, which represents a partnership between Radcliffe, the Arboretum, and the Graduate School of Design. So that's one of the things we have coming up. Now we have a public reception downstairs on the first floor of Knaufel. We hope that you will join us, and perhaps we can applaud all of our speakers here and those who are still in the audience. Thank you all very much.